my degree, my master's was in molecular biology, like you have. And I did my PhD in medical oncology, but I'm fellow of Max Planck in the field of infection biology. So you will be asking yourself, how come actually? How come from cancer to infection? So that is the beauty of the science, particularly the molecular biology, that you can have links and you can have many links which you cannot or someone cannot on the first hand cannot even imagine. So I tell you, I give you my talk like a story. Don't take it as a scientific talk, take it like a story. So one of you at least know or some of you know about RAS. This is a oncogene, okay, and that was discovered long ago, and everyone know that it can cause cancer if it is mutated or if it is activated. So some 25 years ago, this was 1989, when a Japanese group of scientists, they discovered another brother of RAS. How can I say it is brother of RAS? Because it was highly similar. And when they put this gene into the cell, back into the cell, cancerous cells, it became normal. And everyone was very excited. They thought we have, searched the cause of the cancer, and we can cure the cancer by this protein, and the name of this protein was RAP1. 1989. Then lots of discussion went on in the field, and people started proving or disproving the original hypothesis that RAP1 can antagonize RAS or it can cure the cancer. And some other groups in different countries said that actually it can cause the cancer by itself. So it became very confusing. 1999, a scientist, Asha, she told that actually RAP1 is also involved in the embryogenesis. 2000, someone said that it can activate integrins. Maybe you don't know integrins, but actually these are again some specific proteins which can make the cell move. So all of you know that cell movement or migration is very important phenomenon in cancer. By your experience you know it, or from your education you know it. A tumor cell can become cancerous cell, it can migrate into different places and it can basically enter and spread into a whole body. So migration takes place, right? And someone discovered that this migration is under the control of the protein RAP1. Then another group of the scientists said that it can even affect the adhesion. What is adhesion? Attachment. So how it is important to the cancer? That when we have tumor, the tumor cell can have less attachment and it can start migrating. So more attachment, it won't migrate. So when it become like floating kind of cell, so RAP1 can also control the attachment. And means this was how many years? More than 20 years almost gone, but no one knew how RAP1 function. What is upstream to RAP1 and what is downstream to RAP1, right? So for the first time in 2003, when I started my PhD, again a Japanese group, Katagiri et al., they discovered a protein, they gave it a name RAP-L, and they found that it is downstream to RAP1, and RAP1 can control some of the functions which I mentioned before through this protein. So first discovery in the field of RAP1 after 20 years. But the problem here was, this was a very good paper and published in I think Nature Genetics or so. It's quite good journal. The problem here was that all the functions of RAP1 like embryogenesis, adhesion, migration could not be explained just by one pathway. So it means there were many other pathways which were unexplored. So second downstream target came in 2004, and they said that through RIAM, another protein, RAP1 can control the adhesion. And then there was nothing. So during these 20 years, people also worked how RAP1 work, and I won't go into the complications and of the scientific difficulties and the complexities over here. But scientists discovered that RAP1 can work actually like a switch. It can become on, literally it can work like a switch. It can, like electricity switch, it can become on, it can become off. And when it become on, it become active and it passes the signal. And when it is off, it doesn't pass the signal. And the on-off mechanism of RAP1 is controlled by its binding to another molecule. Its name is GDP. and GTP. When it bound to GTP, it become active, and when it GTP hydrolyzes into GDP, it become inactive. A domain in RAPL protein, which is already a known partner of RAP1, and theoretically also, because this is highly similar, and you can see the residues, positively charged residues, K R, K and K, they are conserved in both the proteins, so they are responsible for interaction of protein 1 with protein 2. So and just first and very quick and dirty experiment we did was we overexpressed this RASSF1 into the cells using the plasmid system, and using the GST pull down. Maybe you don't know, but it's very simple. We take the RAP1 protein and we tag it with another protein which has affinity. We just load that RAP1 protein on the beads, okay, silica beads, and we mix the probable partner in single tube and let them interact with each other in a favorable environment, and we just pull it down, we just precipitate it and check. We precipitate using protein 1 and check for protein 2, right? So if they interact, so we will see protein 2 in the precipitate.